Thank you for joining us today for this webinar between PPMD and Wave Life Sciences. I'm joined from PPMD today by Pat Furlong, our President and CEO, and then members of the Wave Life Sciences team. We have Paul and Anne-Marie on. They're going to talk through Wave's product N531, which is an exon skipping product for those who are amenable to exon 53 skipping. They're going to discuss some of the preclinical data that they've been working on, as well as some early, in-human data, and a little bit about FORWARD-53, their trial that they're designing for N531. We are recording this webinar, so if you have family members or friends who this would be relevant to, please feel free to share this with them in the week following. And then we will have an open Q&A at the end of today's webinar. So if you have any questions that come up while Wave is discussing the presentation, please put them into the chat below, or questions that come up while we're discussing it. And we'll try to get to as many of those as we can. So with that, I'm going to hand it over to Paul to take us away today.
Great. Thank you, Eric. And thanks, Pat, as well, for this opportunity today. And thank you, everybody, for taking time out of your busy days to join us for the webinar. A quick introduction to myself and then Anne-Marie. So I'm Paul Bolno. I'm the CEO of Wave Life Sciences. And I've been in this position at Wave for now over a decade. And we'll share a lot about where the company has traveled in that time with the community as partners. Anne-Marie.
Hi, everyone. My name is Anne-Marie Lee Kwai Chung. I'm the Chief Development Officer at Wave. I'm a chemist by training and a 20-year-plus veteran in drug development. Only 2 years with Wave, but hoping to be here for a long time. Thanks, Paul.
We'd like to have you here for a long time. If you go to the next slide. It is important because we know that not everybody is familiar with Wave. So for those not... There we go. For those less familiar with Wave, we're a biotechnology company focused on unlocking the broad potential of RNA medicines to transform human health. And I think if we think about that statement much more broadly, I think lots of companies bring science to the table. I think one important component that we have at Wave, and I'm inherently biased, is an incredible team of physicians, scientists, and members who are really committed to putting patients first. And that's been the hallmark of what we've done at Wave from the very beginning. If we think about the history and my time at Wave over the last decade, it's really been important to take transformational science, this idea of if we have single oligonucleotides, meaning a single medicine, and can learn about what the implication of that is for. And we'll talk today a lot about the exon skipping technology, but we looked at RNA medicines much more broadly and said, how can we drive this forward? When we say putting patients first, it is important to note that our time with the communities that we serve started at the very beginning of Wave and informed of a lot of our decision-making, not just in how we developed our science, but the indications with which we sought to pursue. So really over the first several years at Wave, we were learning about our capability, we were learning about our science, and we were spending time with the community. Our time at Wave is paralleled very much with the Duchenne community, and we'll speak to that relationship, starting really in 2014 when we identified the potential of our exon skipping oligonucleotides to generate functional dystrophin protein. We then are seeking collaboration, so we work deeply with Oxford University and Matthew Wood, really to test and identify the ability to do what we believe would be transformative medicines to increase dystrophin protein, of functional characteristics of dystrophin protein, and to develop an exon skipping therapy leading with our new technology. We started, and some of you may be familiar with our initial clinical programs in suvodirsen. So we initiated that program back in 2017, and we learned through that experience at the very beginning was just how important it was if we were going to transition medicines to patients quickly, having manufacturing capability in-house to be able to accelerate insights that we were seeing preclinically and ultimately transition them rapidly into the clinic. We made that transition, and while there was a lot of growth in the beginning, I think we continued to do a lot of learning with the community as we progressed. And 2019 was a formative year for Wave in that on one hand, we had really come with alignment with the FDA, partnership with the community on ways of expediting late-stage development for a multi-exon skipping franchise, and that was the complex innovative trial design that we launched alongside suvodirsen. But we also had a setback with suvodirsen and stopped that clinical trial. And I actually had a moment to reflect on this, and I don't want to put Pat on the spot, but I do have a moment to reflect on that with somebody who was in that room with me that day. And the one thing that I still take when people say, what was the most influential moment at Wave? It was actually the call before we announced that clinical trial stoppage, where we had the call with Pat and the community ahead of time. And I remember Pat saying to us, don't give up and don't abandon the community. And that resonated with us. It resonated with us because we had done a lot of work in terms of understanding not just how to make suvodirsen, but importantly, really, what does it mean when one stops a clinical trial? Our responsibility to the community and patients to assure that they have their samples, to have their conversations with their physicians, and most importantly, really to understand where the disconnect was preclinically on that translation. And our team went to work. We didn't abandon the community, but we also said it was important for us, if we were going to come back to the community with a program, that we really understand why. Why do we believe that we should be bringing back a program into the clinic? And so the team set at work. And what you see in 2020 was a transformation on the platform, a new chemistry, which gave us better access to muscle, gave us better access to the right compartment in the muscle, and that translated to much higher dystrophin skipping and protein, and Anne-Marie will share that data later. We then took a really responsible next step, which was to say, instead of going back to what we did prior, to run a small exploratory study. And we're grateful to the patients who participated in that initial clinical experiment, which was fundamentally designed to answer those questions of, did the data that we saw now in an animal model translate into patients? Meaning, do we see the reason to believe to move forward? And we shared those data with the community in December of 2022. Then rapidly moving from that point forward, we did have a positive data in 2022 to rapidly pursuing what we will introduce is FORWARD-53, which is our potentially registrational phase 2 study. And Anne-Marie will share more about those details. I think the important part as we think about going forward is we're excited about the progress that we've been making with exon 53 in our FORWARD-53 study. We're excited about continuing to explore multiple exons on the other side of those data and to really continue our partnership with the community as we advance the DMD franchise. We go to the next. So if we think about on this call, the discussion has two main nomenclatures. You're going to hear a lot about WVE-N531, and you're going to hear about FORWARD-53. And so to put those pieces together, FORWARD-53 is the potentially registrational clinical study using WVE-N531. This is designed to demonstrate that we can sufficiently produce functional dystrophin protein with an improvement in exon skipping. Core to this, so when we hear about N531, you'll hear a lot about that nomenclature called PN chemistry. And I know people sometimes go, well, what is PN? And without getting into a deep scientific chemistry lesson, I think the key on PN is you should think about it as a novel modification on the backbone that doesn't have us having to put conjugates. So we don't have to deliver this with conjugates or antibodies or proteins, but rather changing the constitution of the backbone of the chemistry really lets us modify how that medicine's delivered, how it gets to the right compartment inside the cell, and ultimately how that translates to producing exon skipping and functional protein. And with that, I'll transition to Anne-Marie, who will go into more detail about the data that supported this transition.
Thank you, Paul. So as Paul outlined, we took important learnings from the suvodirsen program and translated them into our N531 program. So in the next few slides, I'm going to explore in a bit more detail the data that supported this transition, how it translated into the clinic, and where we are today. And so starting with the preclinical data, a really important step in any research program to move into clinical studies is to determine the potential for benefit. So we start by trying to determine whether there are pharmacodynamic effects. In DMD, you may be aware there are some important mouse models that help us understand the pharmacodynamics. A transgenic mouse is a mouse where the mouse DNA either has a spontaneous mutation, like the human disease, or has been altered to carry the mutant gene. And the MDX mouse has a spontaneous nonsense mutation at exon 23. This means that the mice are not able to express full-length dystrophin. They are still able to express utrophin, and utrophin is a smaller protein that's upregulated to compensate for the lack of dystrophin, which can attenuate muscle damage. So despite not being able to produce full-length dystrophin, the MDX mice have minimal clinical symptoms. Their lifespan is only moderately reduced, and this means that we can't look at whether an investigational drug is having an impact on the disease phenotype. On the other hand, the dystrophin-utrophin double knockout mouse has been engineered to exhibit symptoms like what we see in human disease, muscle degeneration, cardiomyopathy, and an impact on, unfortunately, lifespan. And this means in this model, we can determine whether the investigational therapy is impacting the disease phenotype. And this is what we used to assess N531. And the data gave us significantly more confidence on the potential for N531. So if we move to the next slide, I'm showing here the outcome of the experiment in the double knockout mice. And because the model is an exon 23 mutation, we used an exon 23 targeting molecule with the same chemistry as N531. And we compared it to an exon 23 targeting suvodirsen molecule. We dosed the animals every other week. And in the left panel, you can see the impact that this had on survival. The red arm over there to the far left is the control arm. And these mice had a mean survival of about 7 weeks, which really speaks to the severity of this phenotype in this animal model. The blue arm is our suvodirsen comparator. And these mice had a mean survival of 12 weeks. And across the top, you can see a green line. And if you squint a little bit, a dotted black line. The green line is a 75 milligram per kilogram N531. And the dotted black line is 150 milligrams per kilogram. And in these two arms, there was a median survival of 40 weeks, at which point at the end of the experiment, the animals were sacrificed. So we saw this huge impact on survival in this animal model. And in the right-hand panel, in addition to that impact on survival, you can see how N531 impacted function in the model. So N531 restored muscle and respiratory function equivalent to that of wild type. There's a black line and a blue line that you could pay attention to. And so these data were truly encouraging to us as we moved further into understanding the properties of this molecule. Next slide, please. So here we are looking at a bit more into the double knockout mouse. And we compared the tissue concentrations of the suvodirsen versus the N531 molecule. And we could see that the N531 concentrations were significantly greater than suvodirsen. And in addition to that, greater in the heart and diaphragm compared with the skeletal muscle, which is, of course, where you need them most. And we saw exon skipping in this model and significant increases of dystrophin in all tissues at the 150 milligram per kilogram dose. Next slide, please. So on the left panel, in non-human primates, we saw N531 concentrations that really echoed what we saw in the double knockout disease model. And with significant concentrations in all the tissues, including, as before, higher concentrations in diaphragm and heart compared with skeletal muscle, and at doses equivalent to what we are studying in humans. And on the right panel, this is an in vitro test which assesses the ability of N531 to produce dystrophin in patient-derived myoblasts. The N531 is cultured with myoblasts in free uptake conditions at 10 micromolar, and we use a wild type control to create a titration curve so that we can interpret the results that we see. And we saw concentration dependent dystrophin protein restored across the doses. So these data were extremely encouraging. and indicated that the PN chemistry utilized by N531 had significantly improved our chances to produce dystrophin. And with that, as Paul mentioned, we initiated a small study in humans to really test the hypothesis. We were really cognizant that we wanted to see translation of adequate concentration of drug into the tissues, localization of the drug in the right cellular compartments, and skipping before we conducted a larger and longer experiment to determine whether N531 could increase dystrophin expression in boys with DMD. So the Part A study was a proof of concept study. And on the next slide, I'm showing the design. So in 3 DMD boys with mutations amenable to exon 53 skipping, we undertook an intra-patient single ascending dose escalation up to the target dose of 10 mg per kg, which is equivalent to the top doses that we used in the animal studies that I just showed you before. There was a washout period once the patients had reached the 10 mg per kg level of up to 2 months. And then we administered 3 consecutive doses of 10 mg per kg every 2 weeks and undertook a muscle biopsy 2 weeks after the final dose. And dystrophin is a large protein, and its expression can take some time to manifest at significant levels. So we weren't expecting to see dystrophin after just 3 doses of the drug. Next slide, please. So encouragingly, N531 appeared to be very well tolerated, although of course this is 3 patients, but all the treatment adverse events were mild, with the exception of a moderate COVID infection. This was during the pandemic times. There were no serious adverse events, no stopping criteria were met, and there were no discontinuations. And importantly for us, N531 was much better tolerated with respect to infusion reactions compared with the previous molecule, suvodirsen. Next slide, please. So now I'm happy to present the data that Paul alluded to from Part A back in December 2022, where I think we finally delivered on this long journey that we'd undertaken from stopping suvodirsen to today. And in this part of the study, we saw really substantial tissue concentrations after just 3 doses, 42 micrograms per gram. And this is on a par with what we saw in the double knockout model. And note that that figure is in micrograms per gram. Sometimes you'll see other developers express their concentrations in nanograms per gram, and that is 1,000-fold lower concentration than what you see here. So we went from having no skipping with suvodirsen to a mean of 53% exon skipping, and our half-life has improved from 18 hours to 25 days, and that means that the drug can really be located in the right compartment for even longer, and it gives us much greater opportunity for PD effects. On the right-hand side of this slide, you can see a patient biopsy from Part A. This is just 1 patient, but we saw similar results across the 3 patients. So in healthy conditions and with dystrophin, stem cells divide in response to injury and create new myocytes. But in the absence of dystrophin over time, the stem cells don't divide properly and there's a reduction in regeneration of the muscle. And in this picture, if the biopsy, the brown dots are the nuclei of the stem cells and the red staining is N531. So we're really encouraged to see a clear uptake of N531 in these stem cells, which could have some interesting implications for muscle regeneration. And to our knowledge, this is the first clinical data that shows uptake in stem cells. And finally, the PK data from this study suggests we will be able to have bi-weekly or possibly monthly dosing, which could really significantly benefit patients and their families in the burden of being treated for DMD. Next slide, please. So I'm going to talk a little bit more now about the FORWARD-53 study design, which is our next step in our investigations into N531. FORWARD-53 is an open label study. It's potentially registrational. We're studying 10 boys with exon 53 amenable DMD. 10 mg per kg doses are administered intravenously every 2 weeks. And we're planning to take biopsies at 6 months and 12 months to determine whether dystrophin protein levels have been restored. And additionally, we're looking at functional assessments and, of course, safety assessments as we undertake the study. Sorry, next slide, please. The study is fully enrolled and we are progressing well through dosing. And we're expecting 6-month data, the 6-month biopsy data in Q3 of this year. Next slide, please. In designing the study, we're really grateful for the support of the community and the feedback the community has given us. And we recognise this as a huge commitment to be in a study. It has a lot of impact on the boys and their families. And so we've taken some measures to try and limit that impact on them. We opted for an open label study design to reduce the need for placebo. And we've limited the number of biopsies. Obviously, these are invasive procedures with a lot of consequences for patients. We introduced a concierge service to try and decrease inconvenience for patients having to travel to the site and out-of-pocket expenses that families have to bear in order to get there. And as the trial progresses, we will be introducing home nursing to limit the amount of time that patients and their families have to take to complete study assessments. So we're excited about the study and we look forward to keeping you informed about the programme and sharing data with you in Q3. Next slide, please. So just in summary, I think our journey in N531, it tells a really important story about how scientific progress works. Every experiment, regardless of outcome, brings the community closer to an effective treatment for DMD, and having to stop the suvodirsen program and take those learnings and optimize our animal studies, this has really allowed us to understand the opportunity for N531 to have pharmacodynamic effect. And having established in animals that we could achieve high tissue concentrations, exon skipping and dystrophin, and impact a disease model, this enabled us to move to Part A, and with very encouraging data from Part A, where we saw these very high levels of exon skipping, adequate tissue concentrations, and this novel data reaching the stem cells, and coupled with N531 being well tolerated and having a PK profile that could support less frequent dosing, we initiated our FORWARD-53 study. Our FORWARD-53 study is obviously critical to determine whether N531 produces dystrophin levels that could be meaningful for the community and to better understand the safety and tolerability of N531. Next slide, please. So N531 may address a high unmet need in DMD patients, but of course there are many other exons that also will need to be addressed. And we are working on a strategy for other exons beyond exon 53. We've already generated preclinical data to support around 40% of the DMD population. And these follow-on exon skipping compounds, all with the PN chemistry that we use in N531. In vitro studies, we can already see high levels of exon skipping and dystrophin protein restoration. And so this is also encouraging as we think about the future opportunities for other patients. So with that, I'd like to thank you and hand back to Paul.
No, and just to echo that, and thank you, Anne-Marie, for sharing that. And most importantly, thank you all, not just for your participation in trials, but really, as Anne-Marie had mentioned, the engagement that goes into planning for those studies and really what enables us to wake up every day and do what we do. I think the remaining part of the call is really about you. So we want to make sure that there's substantial amount of time for questions. So Anne-Marie and I are both here and both available to answer your questions about our work in DMD and in particular N531 in this study.
Thank you. Thank you, Paul, and thank you, Anne-Marie. This has been really, really helpful. If it's okay with you, Eric, I'll take through some questions before I turn it back over to you.
Sounds good.
So I'm going to go through, and I see the questions coming in online. So I'm going to go through your slides and I'll bring all these questions in. Paul, I'm just going to go to slide 3 again, just because I think that it's really important to note and for you to explain a little bit about on slide 3, when you talked about your previous study with suvodirsen and the complex innovative design. Can you just describe that a little bit for us? Are you continuing with that program, or can you just shed a little light on that?
Yeah, no, and thank you, Pat, for bringing us back to that, because I do think that that was something that, we had heard, and, at that time point, when one thinks about, how to really attempt to do two important things. One was, how do we think about the multi-exon strategy of bringing multiple programs forward into late-stage development? What was so unique about this study was we'd also heard from the community about limiting the number of placebo patients in a study. And how do we do that responsibly, where we can deliver on a placebo-controlled study, but at the same time, make sure that we're maximizing opportunities for boys to go on therapy as opposed to placebo. And so this was in deep partnership with the FDA. It was actually eventually published, this design, with the FDA. And the goal of this study really led us to this, was called an augmented placebo group. And what was super helpful, and we really recognize our partners, meaning other companies within DMD space, which was this idea of, can we get better access to placebo data sets across different cohorts of studies? So what we could do was have a small placebo patient population on trial, and then what we call is, you could think about it as a virtual or representative placebo population from existing ongoing studies. And there was a way of statistically modeling those so that we could assure that we were building a placebo cohort that had similar characteristics and was behaving similarly. So if you think about kind of the deep statistical design, what it enabled us to do in parallel was to have an emerging ongoing placebo group that was minimized by actual boys on study, and therefore we were able to continue to drive that forward. That study received a lot of attention. So to your question on thinking about it on a going forward basis, we very much look at that trial design as an opportunity to continue, hopefully this time now with N531 driving a different outcome. But we are committed, as Anne-Marie was saying earlier as well, about thinking about ongoing strategies too, to ensure, one, that we're advancing N531 for boys amenable to exon 53 skipping, but really now thinking through, how do we take this study design to the next level to be an umbrella design, to really be thinking about accelerating other amenable exons as well.
Yeah, thank you. I really was impressed with that, the complex innovative design. I think that using the placebo controls from other studies to basically match children or match people in the study is very helpful, because it's a contemporary natural history, and that way you limit the placebo. And I think I can speak for the community when I can say, we hate placebos. It doesn't feel fair as time marches on with muscle and its degeneration. So I really appreciate that explanation. On slide 4, you talked about this is a novel compound, right? And you also talked about conjugates. I think, just for the community, we're very familiar with PMOs. We're familiar with PPMOs, but these kinds of chemistries are new to us. So if you could just shed a little more light on the PN chemistry, it would be helpful, as the PN chemistry is not very familiar to this community.
No, and thank you for the question. And I think anytime we talk about things that are new, it's important that we do spend time talking about them. So what we call PN, and I think you can think about, as you said, there's lots of Ps. This is always this phosphorus group. So you have what's morpholinos, as you said. There's a lot of familiarity in the exon skipping space with PMOs. And those are neutral, and they tend to be excreted rapidly, which is why you kind of have to give frequent large IV doses in order to get into the muscle. The evolution of that was phosphorothioate chemistry. So people hear PS, or phosphorothioates. And then it added a way of stabilizing these and getting protein bound, but brought other challenges forward. What's been interesting on the chemistry side for us, and again, we'll separate this from PPMO, so these peptide conjugates, hence the first P, or antibody conjugates. And, we can think about peptides in the way of having a charge to try to get in the muscle, ways of solving for accessing muscle. What was unique about the PN modification is it's not a conjugate, and it's an important thing to get out at the front. So this is not a moiety that we're kind of sticking on to the backbone of an oligonucleotide. it's a way of taking phosphorothioate chemistry off, so that if you think about kind of what we like to think about as titrating pharmacology, how do you build a molecule? How do you do rational design to actually build an oligonucleotide? It's a neutral charge. So we're able to balance this kind of concept of charge and neutrality so that you enhance drug getting into the muscle, you translate it into the right compartment. It binds to proteins, it stays around, it can stay around for a very long time, as Anne-Marie said, 25 days to allow monthly dosing. So it's not rapidly excreted. But the fact that we've actually been taking charge off the backbone, as we also saw from our early clinical safety experiments, had a very different profile than phosphorothioates. So it really enabled us to kind of rationally build a medicine that has high tissue uptake, substantial, as you saw, concentrations over even those that are using conjugates to deliver into muscle cells, folds higher than reported data on what conjugates have delivered to muscle. But I always say it's not just about getting into muscle. There's more important things one needs to do beyond getting in. And that is getting to the right place in the muscle to do skipping, right? You have to get the medicine there. And what we see with PN is better trafficking to those compartments where you produce exon skipping. And the beauty is we can measure that. We can measure that in the animal models. We can measure that in the clinical study. So part A, the value of part A was not just showing that we got concentration, but that was translating to the highest levels of exon skipping seen to date at just 6 weeks, so just after 3 doses. So we saw that translation on the chemistry. And there's another last kind of piece, which is also important. So a lot of times people think about drugs and they call it, how do medicines get in? You know, and in pharmacology, it's called drug in. The most important thing that we see is not just accessing muscle, but it's retaining. It's avoiding that leakage, getting out of that compartment. And so we see high retention, meaning that the medicine stays in the cell, it resists being chewed up. So the body has a great way of coming in and trying to chew these up to prevent them from doing their work. And what we see is it actually, with the modification, resists the body's degradation. So it stays in the tissue for a long time so that it's able to keep doing that work. And so that speaks to not just the half-life of the drug, what we call productive half-life of the medicine, meaning it's there and able to continue doing what it's supposed to be doing over time. What we're going to be able to see in the FORWARD-53 study is the translation of this. So we'll have multiple time points, both at 24 weeks and at 48 weeks, so 6 months and a year, where we can look at that ability over time to not just generate protein, but to stay in and be able to continue to do that. So the chemistry is an interesting modification. Again, it's neutral in charge, so it's actually, we see adding a lot of benefit. And we're seeing that translate. And it's important to note, too, that this is a modification that we've been using in multiple medicines now across 100 patients in different studies, whether it's in the CNS, in our Huntington study, we've used it in our ALS study. So we're using it in our RNA editing study for alpha-1 antitrypsin deficiency. And so it's been in patients both in the central nervous system as well as systemically. So it's being used. This is not the first time it's being utilized.
Thank you. And that gives us confidence in safety when it's used in multiple indications. So I appreciate that explanation. I'm sure everyone listening did as well. And I'm going to skip over the animals because, to be honest, mice and non-human primates are not very much as interesting or of interest as our boys. So I'm going to go to slide 11. Slide 11. And this is just putting you on the spot, and I'm sorry to do that, but I wonder, and I'm sure people out here are wondering. So these 3 patients that were brave enough to take a compound that had not been in, and then stayed on the washed out, which is pretty amazing, and then received the 3 doses and then a muscle biopsy. Are they continued on this drug, or are they in the forward study? Please tell me they're continuing on the drug.
Yes, 2 of the patients from Part A have continued into the FORWARD-53 study. 1 patient decided not to continue for other reasons other than their tolerability of the drug. But it was really important for us to ensure that the patients, the boys who really showed up for us in Part A, would have the opportunity if they wanted to continue in FORWARD-53.
Thank you, Anne-Marie. And just tell me a little bit about the infusion. You're infusing 10 milligrams per kilogram. How long does that infuse? It's an IV infusion. I'm assuming. How long does the infusion take?
In our recommendations to the site for the first infusion, we instructed the site to take their times, up to 4 hours. But in practice, we're finding it takes an hour-ish. It seems to be very well tolerated in the study.
And you didn't have any SAE treatment as serious adverse events or adverse events after infusion?
So there has been a pruritus that was treated with antihistamines and responded and didn't reoccur later.
Okay. So pruritus is just itchiness, and usually we get that from various reasons, sometimes an infusion, and is usually very easily treated, and then it goes away and doesn't come back. Is that right?
Yeah, that's right.
Okay. Let's go to slide 13. And I think you talked about this before, but the fact that it's two things. One, it sticks around for 25 days may enable you to expand this from 2 weeks to 3 or longer, right? And then just a comment about stem cells. I think we're learning more in the gene therapy arena with the question of what gets into stem cells and what does that mean and what happens to those stem cells. So since you've demonstrated here that the molecule gets into the stem cells, could we assume, or is there a possibility that not only as this drug is utilized, that it could help with regeneration of muscle and help protect and build muscle? Can you comment on that?
So that is what you would hope could happen, or want to be really careful not to raise expectation, because of course we need to study this to determine whether it would have that effect. As muscle becomes fibrotic and there are fewer stem cells, then there are fewer available stem cells to create myocytes. So it would potentially have a better impact if you could introduce it as early as possible. And that raises the important question about intervening earlier in the disease course. And of course, newborn screening is a really important topic in this community, and a really something that's really moving forwards in a lot of different countries, the US and Europe. And so I think, if these data turn out to be giving the opportunity for regeneration in muscle, it will be important to study patients earlier. But certainly, as we're thinking about the design for our next studies, we're really trying to think about how we could best investigate this further, and how it might show up then in clinical outcomes. And that's something we'll be engaging the community with as we further our design.
I think just to
thank
follow
you.
up on that.
Sorry, Paul.
No, it's okay. Beyond just the stem cells, as you were walking through a little bit of that data, I think what's important too, and I know we jumped over the animal experiments because I hear you, and I think it resonates with us, that at the end, this is about treatments for patients. But what was interesting for us to look at from a data perspective was the concentration and skipping, all of this being in skeletal muscle. And I think we do go back, because I do think it's a substantial point of differentiation for that chemistry question you were asking of, well, what's unique about, this PN modification is we did see, which is different, a much higher level of drug concentration, skipping, and protein in the heart and diaphragm as opposed to skeletal muscle. So of course, in the clinical trial, we are not doing, to be very clear, cardiac biopsies and diaphragm biopsies. But it is important that we're using the muscle biopsy in a very meaningful way, not just to look at how we're doing in skeletal muscle, but it does inform us on modeling where we believe we could be seeing that higher levels of dystrophin production in heart and diaphragm as we think about prevention of cardiomyopathies and respiratory decline. So I just think it is important as we think about this. It is very important.
Yeah, no, I think it's really important at the target tissues, right? The diaphragm seems to be a more difficult muscle to penetrate as the intercostals are, because of, exactly. I think the fact that those are very thin muscles, if you will, for lack of a better description of those muscles. Just while we're talking about penetration, it's hard when we, as a community, see data from various antisense or from gene therapies, and it's, you know, it's described as you have described it here. And to try to compare and contrast results. Well, what does that mean in terms of dissemination? So as I know you're not comparing it to others, and I don't need you to do that here, but I think that at least from the data that I've seen, this is a pretty significant concentration that I've seen in published or otherwise. Does it cross the blood-brain barrier to your knowledge? Do you have any idea about that?
There's work we continue to do, and it's a great question, Pat. That suggests there's opportunities to explore that, and I think PN has gotten us there. To date, we have not explicitly studied that with this clinical candidate, but it's something we are looking about at our chemistry and subsequent chemistries that could potentially be designed to do that. But to date, with N531, that hasn't been something that we've specifically looked on to share. But yeah, I do think you're pointing out something that we're excited about, which is the opportunity to look at that beyond. But thank you too for pointing out the differences on concentration, because you are right. This is folds higher than what's been demonstrated across other published data sets. The nice thing is people have been publishing data sets, so people can go back and do looks and comparisons. We've tried to create, sometimes we have found, so I appreciate Anne-Marie going into the difference between a microgram per gram versus a nanogram per gram, and we're not going to put that math on anyone in the community to have to do, because I've been in meetings where people have said, but you know, that looks lower than what other people have shown before, and we have to refer them to units, because it is a 1,000-fold different when you move across units. And it is an important notion when you are evaluating other programs to really pay attention, as I know you help do for the community and others, to really look at data across comparisons, because it can be difficult to do those comparisons.
Thank you. So just a little plug, our amazing care team has done a brain meeting some time ago. It feels like eons ago with the pandemic in the middle, but Molly Coven and our care team has just published I think it's called the BELLS. Really, it's an evaluation of some of the behavior issues and some of the brain-related issues in Duchenne. So it will soon be in print, and that way that might be a nice tool for you to think about going forward and work with us on that. So that was just a plug. Okay, now going into slide 14, and we'll talk about the study you're doing now, FORWARD-53, maybe 15, sorry. So this is 10 boys, 2 biopsies. Can I ask you the age range of the boys that you've included in this study?
We enrolled boys from 4 to 18, and we enabled patients who were non-ambulatory to join the study as long as their ambulation hadn't been lost in greater than a year. So in the enrolled population, we do have 2 non-ambulatory boys.
Thank you. And so therefore your functional assessments are probably the North Star and the PUL, or have you included others?
That's right. And we've also included the 95% stride velocity using the SysNav device. This is producing some interesting data in other studies, and it's something we wanted to explore further, and it could be important in Europe.
Yeah, no, I think that device is really important for us to learn, especially because the EMA has allowed it in as a primary, and I think we're grateful behind that. And it would be great to use that device, because it certainly is an unbiased report, if you will, of what's happening in terms of stride velocity. So thank you. Do you have cardiac endpoints in this? And what are they?
We do, but I think it's important to say that in this population, none of the boys enrolled have, baseline cardiac problems, and you wouldn't expect them to at this level of their disease progression. Therefore, it would be very difficult to see, any change, because thankfully they're not worsening at this point. But we are looking at cardiac MRI where patients are amenable to do that, or if not, ultrasound.
That's music to our ears. We do appreciate the cardiac MRI. And then this will read out in Q3, which is very exciting. And I'm making an assumption that positive readout would indicate a submission to FDA based on approval based on the surrogate. Is that right? Based on the expression of dystrophin. Okay,
That's
very exciting.
Right. as things stand, that's still a really interesting and important route for bringing new therapies to patients.
And it feels, I mean, I know time is everything for our families, but this has moved rather rapidly. So that's very nice to see that because in this case, an open label study, so there's no placebo and the data coming in after the 48 weeks. So it feels faster somehow. So that's really, really good. And on slide 16, just a comment, thank you for listening to us. I know that probably we harass everyone and including you about biopsies and about travel support and prepayment and the placebo issues. But thank you for really listening to this community and understanding the difficulty, sometimes the challenges to participate, and certainly for the young men participating, their efforts and their willingness to do so. So on your last slide, and then I'll turn it back over to Eric. And this was slide 18.
I'm sorry, I missed your question.
Yes, slide 18. No. So, I see that your portfolio is 51, 44, 52, 45. Those are all very great exons. You know what I'm going to say next. There are people with more rare exons that feel very much left outside the bus, especially when we look at an approval of gene therapy, which excludes 1 to 17, and then after exon 58. So I just want to say to you from our community, 2 things. One is we really need those rare exons to be in the portfolio. And if there's anything we as a community can do to make that happen or help you, we would. And then the last question is that I'd like both you and Cole to answer is, how do you see this, or do you see this alongside a gene therapy for people who are amenable to the skip?
So I think to take both of those, thank you for recognizing that it doesn't stop where the arrow stops. I mean, I think the opportunity we have, and I think really when we say this is a platform capability, is with the chemistry, it is portable across other sequences. So our ability to establish a regulatory precedent on dystrophin, safety database on the chemistry itself, absolutely enables the opportunity to be thinking about, as you said, in partnership with the community, how do we explore and address rare exons and be able to continue that arrow across the circle? And I think we've got a real sincere opportunity to do that, particularly given the trial designs that we're building. For your last, I mean, I think there is always an opportunity for combinations because, gene therapy works differently, right? Gene therapy is a different protein. And so I think there are opportunities we have in thinking about, being able to sustainably deliver functional protein in a way where we can continue treatment and restore protein levels. And I think we've seen in other indications, SMA being a good example, where gene therapy and protein, in that case, Alexima and Spinraza, have coexisted. So I don't think it's an either or. I think as we look for the clinical trials and the clinical progress, it's important to recognize that we are focused on patients who've not been treated with gene therapy because we do need and recognize that in that study, there are various safety events. And it's important that we don't conflate those events and, impact the development of the product. So it is important for us to really run this experiment in a way where we're seeing the maximum potential of exon skipping. But again, in this setting, in real life practice, there is always complementarity across modalities and different mechanisms. Emery, I don't know if there's anything you want to add to that.
No, I think you really covered it well. Thanks, Paul.
Anne-Marie and Paul, I'm going to turn this back over to Eric, but thank you so much for today. Thank you so much for what you're doing, and we'll continue to ask you about those rare exons every time we can.
Please do.
Thank you. Back to you, Eric.
Thanks, Pat.
Thank you, Pat. And I do want to echo Pat's sentiments. I mean, we think it's critically important and glad to see that you're including cardiac MRI in these studies. Excited to see that, preclinical data shows efficiency in getting into the heart. And of course, we want to see kind of get the outcome from that over time with patients. We did have one other question come in just to get your thoughts on, and you probably don't have enough patients in yet, but if you would expect different outcomes as depending on what you're skipping in, depending on the patient's mutation, you might have different dystrophin isoforms essentially being created by your skip. So it might be only missing one exon, it might be missing multiple exons. Do you expect differences or different responses in terms of outcomes for those patients?
I think what we've seen with other things on the market is that is something you do need to closely look at, and in any case, response can be variable between patients on an individual basis, and whether that's being driven by the exons or other factors is difficult to say. But we absolutely will be looking very closely at that when we have our data, and hopefully we'll be seeing significant levels of dystrophin and then digging down to see what the variability, if any, in patient response there could be according to exons.
While it was a small N, I think what's important is we saw very little variability across the mutations in the Part A study. I know we also, when we see that, say it is a small sample size across 3 patients. But I think that was encouraging as we think about it within the category of a single exon and skipping. But importantly, and I think this is the work that the team's really done in building the molecules to address our subsequent exons, is we are seeing dystrophin protein at or above where we are with exon 53. So I think it's important as we sought to move forward in other programs and recognizing that this is the first, that we're creating kind of a benchmark with exon 53 programs such that subsequently, at least based, and obviously this is all in preclinical data, but based on preclinical data, the protein production is the same or higher in the subsequent exons beyond the exon 53 program. And so again, encouraging as we think about the other mutations that it's not trending the other direction. So I think interpatient variability plus cross-exon variability, I think we've taken that very much into account as we look at the preclinical data going into these studies.
Great, thank you. And I think that's the kind of it with the questions that we had. So I do want to thank you both for taking the time to join us today to talk through all of the work you've been doing and the results and where you're headed. For everyone who's listening in, again, this is recorded and we will post this onto PPMD's website. So please let any family or friends who this is relevant to know about the webinar. And then if there are questions that come up, if you're watching this back later, or if you were watching today and then something kind of pops into your head, please feel free to pass that along to us and we can try to get you an answer from the team at Wave. But yeah, that's going to be it for today. So thank you everyone for joining. And again, thank you to the Wave Life Science team for joining us today.
Thank you, Pat, and thank you everybody for taking your time today.